Good evening, this is Mr. Wintermelon. In this lesson, we are going to learn our first type of cell division, which is mitosis. So, by the end of this lesson, you should be able to explain the need for the production of genetically identical cell. The second learning objective is the more important one here, which is to identify with the aid of diagrams the main stages of mitosis. Please take note of the objective. It is to identify the stages of mitosis from diagram. This means they will give you a picture, then you state, oh, this is which stage or which phase of mitosis. You do not have to write essay describing the stages. You just have to identify, point out which stage it is. Look at picture and write down which stage it is. You do not have to copy much of the text. You just need to register the picture in your head. That's about it. Let's recap. The end result of mitosis is one cell will produce genetically identical daughter cell. Why is this important? Because it will allow the cell to function normally by preventing abnormal protein production as well as preventing cancer. This is basically, don't try to fix something that is not broken. When the original cell is working properly, you do not try to modify it, because if you modify it, things might go wrong. So it is important for the daughter cell to be genetically identical, not just to each other, but also to the previous cell where it was divided from. Before we go into the stages of mitosis, there is something that you have to take note, which is called the interphase. This is not part of your syllabus, but it is important to take note that the chromatin replication is done during the interphase. What is interphase? Interphase is basically the phase or the stage where the cell spends most of its time. Interphase consists of mainly the time the cell is growing, and during this period, the cell will undergo DNA synthesis. And when the cell obtains enough nutrients and the signal and condition are right, it will indicate that, oh, I want to divide. So it will undergo DNA synthesis, whereby the chromatin replicate itself. Replicate means making a copy. So your 46 chromatin, they will replicate. That means make an identical copy of itself. When this happens, we will get our sister chromatid. So 46 chromatin will replicate, then we will get 46 pair of sister chromatids. But just bear in mind, each pair of sister chromatids is considered one chromosome. Before we go into the stages of mitosis, let us look at all the 4 stages. Now, I will give you 4 phrases, and please try to match each of the phrases to each of the stage. So which one looks like the first stage? First. Which one looks like the sister chromatid are meeting up? Which one are the chromosome being pulled away? And which ones looks like they are getting tear apart? So I would like to use these 4 phrases to remind me of the stages of mitosis. The 4 stages of mitosis is called: the first stage, the prophase. The second stage is called the metaphase. or the metaphase. The third is anaphase, and the last is the telophase or telophase. So remembering the phases will help you to remember what is going on in the cell for each of the stages. So let us watch a video on the stages of mitosis. The first stage of mitosis is known as the prophase, or what I like to call prola first. So pro is the first phase, and pro start with P for pedestrian. This chromosome, they are standing around like pedestrian, 路人甲,路人乙, not doing anything. So prophase. The next phase is more interesting. It's called the metaphase, or what I like to call the metaphase. So during the metaphase or the metaphase, the sister chromatid, they will meet up in the middle. And this organelle called the centriole will help to arrange them in the middle. Of course, this one, my chromosomes are a bit too long. Okay, so they will meet up in the middle. This is the metaphase or the metaphase. The third phase is known as anaphase. A for anaphase, A for away. Because this is when the sister chromatid gets pulled away from each other. So just take note, when the sister chromatid are combined as one, when they are joined together, they are known as one chromosome. When they get pulled away during the anaphase, the sister chromatid become individual chromosomes. So now we have one chromosome, one chromosome. But when they are together, like this, it is one chromosome. But once they get pulled away, they became individual chromosome. So the centriole, besides arranging the sister chromatid, they will also help to pull the chromosome away to each end of the cell. So once the centriole has put the chromosome to each end, the next phase will begin. It's known as the telophase, or I like to call it the telophase, because the cell gets telo into two separate cells. But the term is known as cytokinesis. So the cell will separate or get tear into two or get divided into two, and we have the end result of mitosis. So we compare these two cells. Let's compare these two cells. Okay, let me just put the chromosome. So now we have two separate cells. We can see that we have 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 6 chromosomes in each cell, and each of the cells have 3 sets of chromosomes. Do we have 3 sets? Yep. So the end result of mitosis is you will have genetically identical cells. Now that you have learned about the 4 stages of mitosis, let us just briefly summarize what you have learned. So the first stage is called the prophase. What is not shown in the video is the spindle fiber. Centriole, which was represented by the paperclip, will still be there, but this spindle fiber was not shown in the video. The function of this spindle fiber is to arrange the chromosome into their respective places, as well as pulling them away from each other during anaphase. So you can see centriole like the puppet master, then the spindle fiber, like the string that is attached to the puppet, which is the chromosome. But again, spindle fiber and even centriole is not very important. Spindle fiber, I have never seen them mentioned before in O-level. So don't worry about spindle fiber. It is more of just for your info. They are there to arrange the chromosome into places. So we can see some description of prophase. If you want, you can just read them up. But what is more important is you being able to identify that this picture, the cell is in the stage of prophase, whereby the chromosomes are just sitting around like pedestrian minding their own business all over the place. This is prophase. Second phase is the metaphase or the metaphase. And as before, it is more important that you are able to identify that this picture, the sister chromatids are meeting up in the middle. So the sister chromatids align themselves along the metaphase plane. This is the metaphase plane. You do not need to know the name. You just need to know that they align in the middle. In the middle of the chromosome is the centromere. They are attached on both sides to the spindle fiber, so that later on the spindle fiber can pull them to opposite end. But again, you do not need to know this description. It's here in case you are wondering how the sister chromatids get pulled apart. The next stage is the pulling away phase. A for away, A for anaphase. Again, you should be able to tell from this diagram that the sister chromatids are getting pulled apart in this diagram. So during anaphase, sister chromatids will be pulled apart to opposite end. Once they are separated, right, they are called daughter chromosomes, or they are just called chromosomes. You do not have to worry about the word daughter. So in this example, before they are pulled apart, there are 2 chromosomes. After they are pulled apart, there are 4 chromosomes. It is something like your 油条 or your 油炸粿. If you order 1 油炸粿, they will give you 1 油炸粿, which is like 2 油炸粿. When you go and pull your 1 油炸粿into 2 油炸粿, you will get 2 油炸粿. So this is the idea of how the sister chromatids, which is 1 chromosome, became 2 chromosomes when they are pulled apart. Finally, the last phase is called the telophase or the telophase. So during telophase, you can see that the chromosomes are at both pole or both end. Other description is not important. And as mentioned, the cell will undergo cytokinesis, whereby cleavage will start to appear and the cleavage will deepen. And the end result is we produce 2 genetically identical daughter cells. So this will be all for mitosis. The important thing is you need to be able to identify the phases from diagram, and that's all.